Welcome back. In this session, we will discuss on the epithelial tissue. Do you know what is histology? How do we observe the details of any section? And what are histological sections? Before all this, why should we know all these things for discussing epithelial tissue? We want to discuss about the epithelial tissue, right? So, why should we know about this technical part? Hmm? Epithelial tissue is a tissue by itself, but it is always associated with organs or parts of the body. To identify the epithelial tissue, we have to observe under the microscope. It is not visible to the naked eye. Hence, we want to discuss the details regarding histology. So, what is histology? It is the study of microscopic sections of tissues. How do we observe the sections? The details are observed in a section by focusing it under the microscope. What are histological sections then? They are thin, flat slices of tissues or organs. Okay, thin slice of the tissue is taken of any part of the body, okay, which undergoes a series of processes during preparation of slides. Okay, so it is fixed with 10% formalin, dehydrated with different grades of alcohol, embedded in paraffin wax. Then blocks are made with paraffin wax, okay, where the tissue is embedded in the paraffin wax and a wax block is made. Okay, so after the wax block is made, thin slices of the tissues from this block are sectioned, okay, with the help of microtome. Then these sections are processed, okay, they are placed on a glass slide, they are processed, then stained, okay, with hematoxylin and eosin, and then the section is sealed with cover slips. Okay, so this slide which is now ready will be focused under the microscope, okay, to view the details. So by the end of the session, okay, in this session what are we going to cover is you will be able to understand the principles of histology, outline the types of epithelium and give suitable examples, okay, describe the functions of epithelium. Okay, so here in this particular session we will go through the first learning outcome, okay, that is understanding the principles of histology. The other two outcomes, okay, will be dealt in the coming sessions. Okay, so this picture shows the microscopic slide onto which the thin slices of tissues are placed and processing is done. Okay, and the cover slip to cover the tissue after processing. The lower picture, it shows the mounted sections after processing, which is ready to observe under the microscope for identifying the features or details. Okay, this is how the slide looks after all the processing is done. Okay, so this is now ready to view under the microscope. Okay, so what are tissues and organs? Okay, these are nothing but aggregation of cells and fibers in different composition. Okay, so the aggregation of cells and fibers in different composition makes up the tissues, and basic tissues together form the organs. Relationship from cells to organ systems. I have been repeating this several times, okay, so that you get the orientation of the different levels of organization of the body parts. So cells, they aggregate to form the tissues. So there are four basic tissues: epithelial tissue, connective tissue, muscle and nervous tissue. So all these tissues together make the organs, and the organs which have particular functions and perform their function together makes the organ systems. Okay. So we can take the histological section either at the level of the tissue or the organ, okay, for processing to view the details. Once the sections are taken and processed, when the sections are observed under the microscope for the details, we should have some idea of how do they look in sections, okay, under the microscope. We need to know generally how do they look under the microscope so that it will be easy to identify the tissue or organ, whether the tissue is normal or pathological. Sections are normally composed of cellular, solid and tubular structures. The cells exhibit a variety of shapes, sizes and layers. Fibrous structures are solid and are found in connective nervous and muscle tissues. Okay. Tubular structures are hollow and represent various types of blood vessels, ducts and glands of the body. Okay, so that is the three types of tissues or organs what we should know in general, so that when we go to the specific tissue or organ to identify, it will be easy for us, okay, when we get the orientation. Are you curious to know how the structures are seen under the microscope? The tissues are present in various forms, shapes and sizes. Now, to understand the microscopic sections of the tissues, we should know the appearance of the structures in different planes. Let us see how it looks for cell, solid structure and how it looks for luminal structure. Okay. So, for any solid structure, it will appear as aggregations of cells without any lumen. Okay. So, here you should observe for the arrangement of cells to identify what solid structure it is. This you will study as we discuss specific solid organ slices, okay, when we observe under the microscope. But we will look into the general appearance of cell and luminal structure in different planes of section. So first let us see for the single cell, okay. The sections look different in different planes of a single cell. Let us get the orientation of it. The size, shape of the cell wall vary in different types of cells and also in different planes of section. When the plane of section goes through midline of the cell longitudinally, you can see the cell is split in two equal halves. When you observe the split part of the cell from the side, the split half of the cell, cytoplasm and the nucleus can be observed. So, in this section where the plane is going in mid plane, okay, which is called as the mid sagittal section, okay, and when we look the section, we can see a large round nucleus, okay, on either side of the section, okay. So, when the section passes little lateral to the mid sagittal plane, okay, so this section we call it as sagittal section. Here you can observe the two parts are asymmetrical, right? You can see the two parts are asymmetrical, okay. Just get the orientation of this. In the previous section, okay, where it passed in the midline, there was two symmetrical halves, okay. Here you can observe the two parts are symmetrical. One is large, okay, which is represented by A, and one is small, which is represented by B, okay. Theoretically, it is important to understand which part we are looking into, if you observe the part A from side, okay, the cell is large and the nucleus is small. Okay, because here you can see the section is passing, okay, over, even the nucleus is asymmetrical here. Okay, when it passes over the nucleus, we get a small part, okay, of the nucleus and a large part of the nucleus. But when we look, when the section is passing, the size, what we look from either side will be same, okay, for the nucleus. So here you can see in the part B, when you look from the side, okay, you can see the small nucleus and the section, okay, because it is passing to one side, okay, the section also is smaller, both for the cell and the nucleus. So when the section passes even more peripherally, okay, over the cell, you can observe the difference in the size. When we observe from the front view, one part is represented by A is very large, okay, very large compared to the other part represented by B, okay, which is very small. You can see this difference. A is very large and B is very small. So when we observe the side view of these things, okay, the part B, you can see the section is not gone through the nucleus. So we don't see any nucleus in this section. So as the section passes towards the periphery, we need to understand in most cases the size of cell, depending on its shape and nucleus, it reduces towards the periphery of cell. Okay. So basically what you need to understand on the whole is the appearance of the cell depends on the plane of section. So you can see what is the difference between the sections passing through different planes, okay, in this slide. From center to periphery, that is represented from A to C respectively, the size of the nucleus reduces from large, okay, to smaller size, and close to the periphery it disappears. Okay. The difference in the size of the cell depends on the shape of the cell in different planes of section. If it is oval, it reduces in size as the plane passes towards periphery. If it is cuboidal or columnar, the size almost remains same. Size of the cell, okay, remains same. But the nucleus depends on what plane it will go through. The same way as longitudinal section of the cell, when we look at the horizontal section, the size of the nucleus is maximum at its center. Okay, when it passes through the mid horizontal section, okay, the nucleus is large. Okay, so as we go towards peripheral planes, the size of the nucleus reduces, okay, as in the longitudinal section. Okay, the same way here also, as we go towards the periphery, the nucleus becomes smaller and it disappears to one end of the cell. Okay, so size of nucleus is maximum at its center, and towards periphery it becomes smaller and will not be observed to the end of the plane passing near the tangential part of the cell. So that is how you identify the cells based on the sections passing through different planes. So now let us see for sections passing through luminal structures. The sections look different for luminal structures. For example, we will observe the plane of sections passing in different directions through a simple coiled tube and try to understand how the sections at those levels look like. When the section passes in longitudinal plane along the tube, the section appears long, okay, with the sides of the long tube lined by the cells with elongated lumen inside. Okay, so that is the lumen, and we can see the two walls of the sections. Okay, so we can see the walls of the tube on either side, okay, with a lumen inside, okay, which is elongated. So the cells can be seen lining the two walls of the cells. When the section passes in transverse plane, okay, through a single point, a small round structure lined by single layer of cells with lumen inside can be observed. Okay, so you can see a round structure, okay, because it is going through transversely or horizontally across the single tube. So we can see a single tube, okay, lined by a single layer of cells, okay, with a round lumen inside. When the section passes horizontally through several points, okay, more than one section can be observed which are lined by single layer of cells with lumen inside in each of the section. Okay, so you can see several cross sections, okay, because it is even if it is a single tube, okay, it is passing through several parts of the tube, okay, because it is coiled, and each section, okay, gives you a cross section, one cross section. Okay, so as it passes how many parts of the tubes, okay, across the coiling, okay, that many sections can be observed. Okay, so this type of section is observed in sweat gland. Here the sections are placed in a line to give the orientation, that is in this slide, but actually in a real tissue section the cross sections will be randomly or irregularly arranged. Okay, when the section passes at a U-turn of the tube, okay, where it is bending, okay, here the two lumens, okay, is joined in between by solid layer of cells. Okay, so that is what you will observe. You can observe two cross sections, okay, with a lumen and a central part, okay, without a lumen, which is connecting these two parts. Okay, so this region here will be the connecting part which does not have lumen. Okay, and this section and this section, what is observed in these two cross sections gives the lumen, okay, to look at. When the section passes at the periphery of U-turn in the tangential plane, it results in solid multicellular oval structure without lumen. Okay, so at this end, okay, there won't be the lumen to see. Here there will be lumen. Okay, the lumen is present at this place. Okay, so this region will not have the lumen. Okay, so in the tangential section you can see only the aggregation of cells. So just to brush up the different cross sections to be observed in different planes of sections. Okay, so that is longitudinal section where you can see a elongated lumen and the two elongated walls of the section, okay, with the lining of the cells. The horizontal section passing through single tube, okay, it gives the single cross section with a round lumen inside. When the section passes horizontally across different parts of the coiled tube, there are many cross sections, okay, to be observed. When the section passes through U-turn of the tube, okay, there will be two cross sections with lumen, okay, with a connecting solid cord of cells, and the section passing through tangentially gives rise to aggregation of the solid cord of cells. Okay. So to summarize till now, we have seen the planes of sections of the cell and the luminal or hollow structure with the appearance of these sections in different planes. In the next session, we will discuss the epithelium in detail. Join my Facebook community, link in the description. Like, share and subscribe. Thank you for watching.